In this topic, we are going to discuss a small part. This is the last point in chapter number 4, pharmaceutical preservatives. We are going to just give an overview of this topic. So let's start with what do you mean by pharmaceutical preservative, and we have to understand other basic terms. Basically, what do you mean by preservatives? So preservatives are the chemical substances used to improve or amplify shelf life of drugs by decreasing or lowering the oxidation of active and excipients and by reducing microbial population. So there are many ways by which a drug product formulation can get destroyed, or its shelf life can be decreased, or it can get deteriorated before its shelf life. So one of the reason is microbial contamination or attack of microorganisms, and other reasons might include not storing it at proper temperature or hydrolysis or oxidation. So you need to select the preservative suitable for formulation. So the selection of preservative varies from formulation to formulation. For example, if there are liquid formulations, you have to select the preservative which is basically an antimicrobial agent, because in liquids there are high chances of growth of active microorganisms, maybe bacteria or fungi or molds or yeast, because the liquid favors the growth of such organisms. So for liquid formulations, you have to select the preservative which acts by the mechanism of antimicrobial action. Okay, so for certain agents, certain active ingredients are susceptible to oxidation due to their inherent structure, due to their inherent nature. They are prone to oxidation. So for such kind of formulations, you will have to select preservative which is basically antioxidant in nature. Okay, so you have to basically select a preservative based on the requirement of that formulation and drug and active ingredient. So that varies based on the mechanism required. So there is another term which is known as preservative capacity. So preservative capacity is a term used to describe the cumulative level of contamination that a preserve formulation can tolerate before becoming so depleted as to become ineffective. Okay, so the cumulative level of contamination, what is the level of contamination that that preservative is able to handle? So that is the capacity of preservative. There are like fixed concentrations at which preservatives can be added in the formulation. So if you add higher concentration, higher amount in the formulation, it will definitely be able to fight against a more level of contamination, but that much high amount is not desirable in the formulation, because preservatives basically, for example, say those which are acting by antimicrobial action, those preservatives can be toxic at above a certain level of concentration. So the concentrations of these preservatives to be added in the formulations are always fixed, and mostly these are very added in the very low concentration, so that those preservatives should not interact with other excipient and lead to destroying the formulation. So we have already learned about what is preservative, why it is to be added, and what do you mean by preservative capacity. Now, preservatives are classified in two different ways based on their mechanism of action, and another way of classification is based on their source. So, based on the mechanism of action, preservatives are of three types: antioxidants, antimicrobial agents, and chelating agents. And based on the source, these are of three types: naturally obtained, and two types: naturally obtained and synthetically prepared. So based on the mechanism, the three types are antioxidants, antimicrobial agents, and chelating agents. We are basically interested in the preservatives, those act by antimicrobial action. Nonetheless, we are going to learn so we are basically interested in the preservatives which act by antimicrobial action. So we are also going to discuss the antioxidants and chelating agents. So let's start with antioxidants. So what is the mechanism of these antioxidant preservatives? Antioxidants are used to reduce the oxidation of active compound and other excipients due to formation of free radicals by using their self-reducing activity in the finished products. In simple terms, in simple words, antioxidant preservatives undergo self-oxidation, and by doing so, they prevent active ingredient from undergoing oxidation. So whatever oxidation trigger is present in the environment of formulation, that trigger will be taken ahead, taken by the antioxidant preservative first. So it will preferentially undergo oxidation, and by that process, doing so, it will preserve, it will prevent active compound or other excipients from undergoing the oxidation. So antioxidant preservatives are basically the structures which are, or the compounds which are like very prone to oxidation. So whenever there is any oxidizing trigger present in the environment, they themselves will undergo oxidation and protect the active ingredient from undergoing the oxidation. So these are the examples. These are the commonly most widely used preservatives, antioxidant preservatives: BHA, BHT, and propyl gallate. BHA is butylated hydroxyanisole, BHT is butylated hydroxytoluene, and propyl gallate. If you observe the structure of propyl gallate, you can observe there are many hydroxy groups present in the structure, and like the more the number of hydroxy groups present, more the susceptibility to oxidation. So if you remember, you had prepared one formulation of glycerite in your pharmaceutics practical. It is known as glycerine of tannic acid. And if you remember the structure of tannic acid, it was similar to gallic acid or propyl gallate. There were like many hydroxyl groups present in the structure of tannic acid. And that is why tannic acid is prone to the oxidation. It is susceptible to oxidation. And if you remember, we are adding two agents, two preservatives in tannic acid glycerine. One was antioxidant based and the other was a chelating agent to prevent the oxidation of tannic acid. That was the example. Now, next category is antimicrobial preservatives. So we have already discussed you already know the chemical agents which are used to control the antimicrobial microorganisms. So antimicrobial agents, you have already learned these agents in the chemical part. So all these agents at low concentration can be used as preservatives. So basically it is added in product to minimize the risk of spoilage and to kill low levels of contaminants introduced during storage or repeated use of a multi-dose container. Now, these agents mainly work by inhibiting the cell wall, cell membrane growth, or other bacterial organelles which may attack the product. Okay, so there are for example, a cough syrup. Cough syrup is basically a liquid oral formulation, and it comes in the volume of maybe 100 ml or 250 ml. So to use that cough syrup bottle, you will have to open that bottle multiple times to take the required dose for administration. And during this tenure, during its shelf life, because of the opening of that bottle multiple times, we might introduce the contamination, or microorganisms can get access inside the bottle and cause the spoilage of that product. So to avoid that, you have to add low levels of these preservatives, which are basically antimicrobial agents. Okay, so what kind of formulations require addition of these antimicrobial agents? The formulations which act as very good hosts for growth of organisms. So these are mostly liquid formulations. For example, solutions, suspensions, emulsions, topical preparations, example creams, injectables, eye drops, etc. So all these kind of formulations require addition of preservatives for their shelf life. Now, examples. The first example is benzoic acid. There are like 3 to 4 examples which you will have to by heart for preservatives. So first example is benzoic acid. It is widely used preservative. It is also commonly used as food preservatives along with the medicaments. So it has very broad spectrum of activity. It is also used as antiseptic at higher concentration. So it basically, you can also observe the structure of benzoic acid. It is very simple. It inhibits the growth of microbes, including molds, yeast, and some bacteria. So as we have seen, benzoic acid is very widely and commonly used because it has a wide spectrum of activity. At low concentration, it is used as preservative. At high concentration, it is used as antiseptic as well. Next example is methyl parabenes. So methyl and propyl parabenes, you all must be knowing. You have used these agents, preservatives in pharmaceutics practicals many a times. So parabenes are again commonly used preservatives for topical formulations, liquid orals, etc. Now, what is the difference between methyl and propyl paraben? So methyl paraben is a white crystalline powder. Characteristic, it has characteristic odor. It is freely soluble in water and alcohol. Please note down. It is freely soluble in water and alcohol. So it is used as preservative and antiseptic in various pharmaceutical preparations. It is also used in cosmetic formulations, preparations which are susceptible to decomposition by microbial contamination. So this is the structure of methyl parabene. So in propyl parabene, this methyl group is replaced by propyl group. Okay, and because of this, what is going to happen? The carbon chain length is going to get increased, and as you already must be knowing, as the carbon chain length increases, its compound solubility in water will go on decreasing. Longer the chain length, lower will be the solubility. So this methyl group in propyl, this is methyl parabene. In propyl parabene, this will be replaced by propyl group, and that is why propyl parabene has lesser solubility in water as compared to methyl parabene. And that is the reason when you are preparing an emulsion, while preparing emulsion, you have to mix two phases. One phase is oily phase, other phase is aqueous phase. So how do you add these preservatives in emulsion? You add propyl parabene in oily phase because it has more solubility in oils, and you add a methyl parabene in aqueous phase because of its higher solubility in the water. And then two phases are mixed to prepare the emulsion. So methyl and propyl parabenes, another class of antimicrobial agents which are widely used as preservatives in liquid formulations. Now next one is sodium benzoate. Sodium benzoate is simply the sodium salt of benzoic acid. So it is a white crystalline solid. It is soluble in water and alcohol because being a sodium salt, it becomes soluble. It is used extensively as food and pharmaceutical preservatives because of its again wide spectrum of action. It is not bactericidal, it is only bacteriostatic agent along with the fungistatic activity. So it is active against bacteria as well as fungi. Next, you have two classes and two examples, alcohols and mercurial compounds. Under alcohol, you have example of chlorobutanol. You have used preservative in one of the pharmaceutics formulations. It is a bacteriostatic agent. And under mercurial compound, you have example of phenyl mercuric nitrate, PMN. So it is also used as bacterial preservative and as a topical antiseptic. So these were the examples you have to by heart under the antimicrobial category of preservatives. So after this, there is this last category of chelating agents. So you have used citric acid as chelating agent preservative in tannic acid glycerine. So again, tannic acid was susceptible to oxidation, and to prevent any metallic impurity from triggering the oxidation, you add chelating agent. So most chelating agents act as preservative and protect the product from by forming complex with metals and by preventing its deterioration. Examples are EDTA and citric acid. So EDTA and citric acid are most widely used chelating agents. So what they do, basically they form complexes with metal ions. Metal ions are present as impurities in most of the pharmaceutical formulations because of the large metallic vessels used in their formulation preparation. So those metal ion impurities are chelated, are complexed, or are sequestered by these agents, for example EDTA, and by forming complex with those metal impurities will no longer be available for destroying or for oxidation of the active ingredient or other excipients. So that is how chelating agent works, and under chelating agents we have only two examples, those are EDTA and citric acid. Now, while selecting the preservative, again you will have to decide what are the needs of the formulation. If formulation is susceptible to oxidation, then you will have to add oxidizing antioxidants as preservative. If formulation is susceptible to microbial attack, then you will have to select antimicrobial preservatives. If it has like danger of metallic impurities being present, then you will have to select chelating agent as preservative. So its concentration also needs to be optimized. It should not be added in a very large amount, because then it will lead to toxicity. It should not be added in like very low amount, because then it will not be effective for the function of preservation. So consideration should also be made about its concentration, its toxicity, its selectivity against specific organisms, and its interaction with other formulation ingredients, etc. Now, the last part of this topic is how to evaluate the efficiency of those preservatives. So, evaluation of preservatives. So, we have previously discussed how to evaluate the efficiency of antimicrobial agents. Now, these tests are only to evaluate the efficiency of preservative action. Now, the evaluation of preservatives has traditionally involved time-consuming tests. There are two tests: pharmacopoeial antimicrobial effectiveness test (AAT) and preservative efficacy test (PET). So these are required for assessment of the antimicrobial preservation of multi-use formulation or multiple dose pharmaceutical products. So what is this PET test? In brief, this test involves challenging a product with a defined number of CFU, colony forming units, of variety of test microorganisms, say bacteria or yeast or fungi, and then enumeration at time 0, and then monitoring the kill survival rate at a defined time intervals up to 28 days. So in simple terms, in the formulation in which you have added preservative, you are going to add, or you are going to challenge that formulation with a fixed amount of microorganisms, and then you will test. So you are going to purposely contaminate that formulation, and then you are going to check the efficiency of that preservative to protect the formulation, or to kill the organisms added, or to overcome the contamination. So you are going to make the reading at time 0, at day 0, and then periodically you will go on determining taking the reading for growth or survival, kill or survival of those organisms for the period of 28 days. Okay, so the challenging organisms, or the organisms which you purposely use for contamination, include these test organisms. It can be Staphylococcus aureus, it can be Pseudomonas aeruginosa, it can be Aspergillus niger, or it can be Candida albicans. There is one correction here. Aspergillus niger is the species of fungi or mold, so this should be here. And Candida albicans is an example of yeast. And these two are bacteria. So you have to challenge the formulation with the species of bacteria, fungi, and yeast, because these are the most commonly contaminating species. The product is inoculated with specified number of each challenging organism. Then the inoculated product is held at room temperature for 28 days, and it is examined by the duplicate plate count method. So test is performed run in duplicate. All results are evaluated in accordance with the tabulated acceptance criteria test protocol. So they have a standardized protocol for the observation of results. So for that, they have categorized all the products in 4 categories. So category 1 includes all the injectables, parenterals, ophthalmic formulations, otic formulations, sterile nasal formulations, and products made with aqueous basis or vehicles. So basically, all sterile aseptic formulations are categorized in category 1. In category 2, they have included topically used products made with aqueous bases or vehicles, non-sterile nasal products, emulsions, including those applied to mucous membrane. In category 3, they have oral products other than antacids made with aqueous bases or vehicle. And in category 4, it is specifically for antacids made with aqueous base. So for each category, they have criteria for antimicrobial effectiveness. So for example, for category 1, for sterile products, for aseptic products, for bacteria, 3 log reduction in 14 days and no increase up to 28 days. If this is observed, then only that preservative is said to be effective or able to protect that formulation. For yeast or mold, there should be no increase from initial count at 14 and 28 days. So readings are taken twice, on 14th day and on 28 days. So there should be no increase at all. So the antimicrobial preservative should be able to kill the challenging yeast or mold species at once. Similarly, there are like conditions for category 2 as well as for category 3 and 4. So it is obvious that the conditions are very stringent for category 1 because the category 1 includes sterile aseptic formulations. So apart from that, there are other techniques which are more complex and time consuming. So these methods include ATP bioluminescence method. These are just examples. Electric impedance spectroscopy and spectropurimetry chemiluminescence. So these are just other examples of the method. So as far as writing answer in the exam is concerned, only this one PT method is sufficient. You can describe this method, and you can give a classification of the preservatives based on the mechanism, that which includes the 3 classes: antioxidant, antimicrobial, preservative, and chelating agents. So that was all about the preservatives. This was the last part of chapter number 4. Thank you.